Fungi are eukaryotic microbes also. They are going to be heterotrophic, meaning they don't make their own food. They have to obtain it from another source. And typically that source is going to be something that's dead or dying. So they are saprophobic or saprozoic. That's referring to that. They do include a number of different organisms. Mushrooms would be one of them. Mushrooms are typically pretty large and we can see them with the naked eye. We also have molds. Molds, you may have seen that on different food that's decaying in your refrigerator before. And then we also have yeast, which are going to be single-celled organisms, and that's what we're seeing down here. Now, fungal illnesses are called mycosis. We do have a lot of fungal pathogens of plants. There's also a lot of fungal pathogens of a variety of animals, humans included. But besides that, we also have a lot of benefits to us as humans that we do get from fungi. And some of the things that would include is that they are the source of some of our antibiotics. They're also an important source of food for us, or they help with the production of food, such as yeast being used for the production of alcoholic beverages and also the production of breads. If we're talking about the structure of fungi are eukaryotes, so they are made up of eukaryotic cells. The eukaryotic cells, though, are going to be arranged into the long filaments, like we see here. So it's like a mass web of filaments, and these filaments are called hyphae. So this is the long threads that make them up. These thin filaments are going to have a really great surface area, and that large surface area is going to help with absorption of nutrients from the surroundings or from whatever it is that they're actually infecting. The fungi, they do have a cell wall. Their cell wall is specifically made of chitin, so that is unique to the fungi. This is one thing that helps distinguish them from plants. It also helps distinguish them from things like the oomycetes, which were things that looked like fungi, but they weren't fungi. The arrangements of the hyphae can take on a couple different forms. We can have hyphae or filaments that do have cross walls in them, so that would be the septate. So notice that these cells are separated by cross walls. And then we can also have some where there's no cross walls. And if there's no cross walls, that's what we call cenocytic or non-septate hypha. So the molds, when we're talking about molds, they do have a filamentous structure. If you just think about what you've seen that might have been covered by mold before, if this is, say, an old strawberry or orange, you see it kind of just looks like this hairy mass. Well, basically, that's the hypha extending all throughout that and then also on the surface. And it may be cenocytic, it may be septate hypha. We can also have yeast cells, which are typically unicellular, and they can start to group together. And when they group together, we get what we call false hyphae or pseudohyphae. Some fungi are dimorphic, and what that means is that they can have different forms or different stages that they take on depending on where they're at. This particular picture is showing us that this fungi can be hyphae some of the time, so long filaments, and then other times, like when it's residing inside the human body, it's made up of single cells. Here we have a typical life cycle of a fungi. Some unique things to point out is that fungi very often are capable of asexual reproduction. If it's asexual reproduction, that would mean that it is clonally propagating itself, so it is producing new organisms just like itself. This can be done through the process of spore production, which we see right here. We can also have some sexual reproduction happening, which is happening over here. With the sexual reproduction, this tends to happen in two stages. We have plasmogamy, which is going to be fusion of the cytoplasm. So if I just write that here, this is fusing the cytoplasm. Sometimes they live like this with just the cytoplasm fused for quite some period of time. It could be a couple hours, it could be months or years. And then later we're going to get karyogamy, which is going to be fusing the nucleus or the nuclei. So there'd be two nuclei and we're fusing them together. So when we're talking about sexual reproduction with fungi, first it's plasmogamy, then it's karyogamy, and the timing of those can be pretty much right there together or they can be delayed. In the end, we get a zygote, which is here. The zygote is going to be unique. and that unique genetic combination is going to allow for the process of natural selection to work on it. Depending on the actual structures that are used in the sexual reproductive process, we do have different groups of fungi that we distinguish. So here's the different groups of fungi. There's actually 5 that are being shown in this phylogenetic tree that we're looking at. If we briefly go through and talk about the groups of fungi, the first one are the Chytриdiomycota, or the chytrids. These are known for their flagellated cells. And so you can see the cells over here that have the flagella on them. That's very unique for fungi, that you do have a flagella on there. And these are most known for causing a lot of amphibian deaths and declines in amphibian populations worldwide. So we can see that down here on the bottom. You've got some frogs there. So the chytrids have been implicated in the major amphibian deaths. Another group is Zygoomycota. Zygoomycota is mainly saprophytic, so they are obtaining their nutrients from dead things, but it's not always going to be something that's dead. We do have the most common bread mold included in this group. So that one's Rhizopus stolonifer, and that's what we're actually seeing on this slice of bread right here. Because it is a bread mold, it is responsible for a lot of economic losses, food losses. We also have a fungi included in this group that does cause necrotizing of human tissue, which means that it does cause tissue death in humans. These Zygoomycota are known for the production of what we call as Zygosporangia. So that is their reproductive structure, sexual reproductive structure, is that Zygosporangium. Basically, you've got 2 mating types that are coming together, and they're going to reproduce sexually with that structure. They can also reproduce asexually, which is what we see up here. This would be an asexual spore that we're getting ready to produce. So both asexual means and then also sexual means. Here's just another look at it, emphasizing that we can have sexual and asexual reproduction. We can see the Zygosporangia over here, which is what the group is actually named from. Notice that we have first plasmogamy, fusion of the cytoplasm, and then later we're going to have karyogamy, which is where the nuclei actually fuse together. Here's another kind of unique Zygosporangia. This particular one, you might see it if you go out hiking a lot in the forest, but it actually can aim the sporangia. This is not a Zygosporangia, but it can actually aim that and point the spores in a particular direction and kind of shoot them out. So it can point them towards a particular light source, a moisture source, wherever they want them to go. Another group of fungi is the Glomeromycota. These we don't really think of as being pathogenic. Actually, these are associated with roots of plants in a mutualistic relationship. Mutualistic relationships means that both organisms are benefiting. So here, if we look at the picture, this would be the plant cell, and then all of this inside, that's going to be the fungi. The plant is photosynthesizing, producing a lot of sugars and carbohydrates that the fungi is going to feed off of. And then the fungi is going to be assisting in absorbing nutrients from the soil and providing that to the plant. The Ascomycota are an extremely large group of fungi. We commonly refer to them as a sac fungi. But this includes things like yeast, unicellular fungi, and then also some truffles, morels. It does include bread molds. It does include some plant pathogens. There are some human pathogens included in here, and it also is going to be a group that has a number of members that have been very important to us for antibiotic sources. Penicillium is one of the most popular examples. It is an organism that we derive penicillin from, and it is part of the Ascomycota group. The Ascomycota can reproduce asexually and then also sexually. The top picture here in blue shows its type of asexual reproduction, and its asexual reproduction is through what we call Canadia. Canadia are little spores. They are produced on this larger structure, just one after another. So it looks like this. This is just kind of zooming out on, or zooming in on the picture that we see right here. And each one of these little spores can germinate into a genetically identical organism. Down here at the bottom, we're seeing the sexual reproduction of this particular fungus. This one, they're called sac fungi because they do produce their spores in a sac. So the sac, that's what we're seeing right here. The sac specifically has 8 spores inside of it. It's always 8 spores. And that is something that is used a lot for the identification of these types of fungi. If you have a fungi that produces 8 spores inside of a sac, we know that it falls into the Zygoomycota group. Here's a look at its life cycle without going into a lot of detail here. Just notice that we can get asexual reproduction through spores up here at the top, and then we've also got the 8 spores produced in a sac, which is really what this particular group is named and known for. The last group of fungi that we're going to talk about are the Basidiomycota. These are probably the ones that are the most familiar to you. These are known as the club fungi, and they include your typical mushrooms and what we call shelf fungus. So this is the shelf fungus right here, and then there's a couple of them. And then you've also got your different mushrooms. Many of these are decomposers, so as decomposers they play very important ecological roles. We also have some that we use as food sources for humans, but we do have a number of plant and human pathogens that fall into this group. Looking at their life cycle, what's significant about them is that they do produce their spores. If it's sexual spores, it's going to be 4 of them inside of what's called a basidium. This is underneath that mushroom structure that you might see. So the mushroom is really housing underneath of it all of these sexual spores. And the Basidiomycota, they do tend to reproduce sexually. The last group to talk about when we're looking at eukaryotic microbes are the lichens. We talk about them here. They're a good example of a symbiotic relationship, but we don't really have any pathogenic examples of lichens. They're not infecting humans and animals, not infecting plants. They are a combination of basically 2 types of organisms. It's some type of green algae or cyanobacterium, and that is combined with an Zygoomycota fungus. The relationship between them is very interconnected. The green algae or the cyanobacterium is going to be located in the algal zone, and this is going to photosynthesize. So since this is performing photosynthesis, it is producing sugars, which are going to feed both itself, and it's also going to be feeding the fungus. And then the fungus forms most of the rest of the structure that we see right here, and it is performing really more of a protective role, but it's also absorbing some nutrients from the environment. This, sometimes it's not looked at as being a completely mutualistic relationship because it's thought that the fungi gets a lot more out of the relationship than the algae actually gets out of the relationship. If we look at pictures of it, I'm sure you've seen some of this in the environment. Lichens are commonly seen growing on trees. They could be living, they could be dead. We also see them on rocks, flat surfaces growing a lot. And they do come in a variety of different colors and overall structures. They can be very flat, they can be more raised up and more leafy-like.